Hello, everybody. Welcome to Oceanography. We're carrying on with our introductory lesson or lectures on life in the oceans, life generally. Life in the oceans is integral to understanding life itself because we're almost certain that life evolved in the oceans on Earth. And the reason we know that is that uh, the early Earth was not protected by an ozone layer, probably had a less effective magnetosphere, and so there was a lot more ionizing radiation penetrating the Earth's atmosphere, and would have potentially, had there been life forms evolving, the ionizing radiation would have extinguished them. So the only place that you could live that would be protected from that was the oceans. So understanding life and understanding the evolution of life relates directly to processes in the marine environment. So last Tuesday rather, we talked a lot about cycles, and we ended up by looking at the carbon silicate cycle, which is a very long-term cycle that involves all of the major components of the different Earth cycles. There's the hydrocycle is involved, and the plate tectonic cycle, and of course, these different factors work together. What's important and interesting about the carbon silicate cycle is the way that the formation and the erosion of carbonate-rich sediments on continents controls the concentration of CO2 in the atmosphere, because CO2 is removed when rocks, particularly granitic rocks, dissolve under rainfall. And that's not exactly what's shown in this cartoon, but that's an important fact to understand. So over time, the formation of mountains and then the erosion of mountain ranges has been an important control on the amount of carbon in the atmosphere. Well, why do we care about the amount of carbon in the atmosphere? Well, atmospheric CO2 and atmospheric methane, carbon dioxide and methane, are greenhouse gases. And I'm pretty sure everybody knows what I mean by greenhouse gases. Scientifically, what we're saying is that these are gases that perform a function called radiative forcing in the atmosphere. Basically, they're lids on the atmosphere that scatter light energy and heat energy and keep it in the Earth system for longer than it would otherwise do, thus raising the temperature of the atmosphere and the surface of the Earth. And of course, this is something that we're all aware of. And so tracking CO2 in the atmosphere and tracking methane in the atmosphere is important for understanding, how the greenhouse effect is working both in the present day and as we look backward in the past to try to understand how the Earth may have developed at different times. Now, one of the interesting things that's, of course, as we know, one of the things that's driving the present day climate change is the release of CO2 into the atmosphere because of our use of fossil fuels, and because we have land use processes that have cut down large amounts of forests and have converted large areas of land use into agricultural fields. And the removal of the forests releases a great deal of CO2 because CO2 is stored in standing stock of the biomass of trees, and cutting down trees then ultimately releases that CO2 back into the atmosphere. Likewise, converting soils into agricultural soils removes changes the topsoil from a carbon-rich material into a carbon-poor material, and with the result that CO2 and methane are released into the atmosphere. So, understanding that process, recognizing that is important. So we find out that when we try to track what happens to the carbon dioxide that we use, it turns out that we can't fully account for all the CO2 that's being released. And this is sort of laid out in this little text slide here. And it says that humanity releases 7.1 units per year, or the units in this case are gigatons of carbon per year. And about 3.2 of that remains in the atmosphere. Think about that. We're increasing the amount of CO2 steadily by these 3.2 units. Two of the 7, dissolve into the ocean. And that leaves a remaining 1.2 units, almost a third of the total release, that we really can't account for. We don't know exactly where that goes. Probably what's happening is that there's more effective uptake into the ocean, but this is termed, this CO2 that we can't account for, this part of the carbon cycle that we don't fully quantify, is referred to as the missing carbon. And so this demonstrates this in a little more detail. One of the possible solutions to missing carbon is the formation of gas hydrates, but that doesn't fully account for all that. But gas hydrates are important to understand, and I'll be talking more about gas hydrates when we look at benthic processes and hydrocarbon seeps, which is an area that I've done quite a bit of research on. And so gas hydrates, just briefly, are an ice-like substance that forms under conditions of low temperature, and high pressure. When I say low temperature, I mean cold, like the bottom of the ocean is cold, 2 degrees, 4 degrees Celsius, but not 0 degrees Celsius, not what we would consider freezing point at STP. And so under those conditions, high pressure and low temperature, methane can be incorporated into a lattice of water molecules to form this ice-like substance. And the ice-like substance, gas hydrates, comprise actually a huge pool of carbon, carbon which is semi-stable or across the globe. And so the formation of gas hydrates may contribute to maybe part of the carbon cycle. And one of the concerns about global warming is the feedback effect. So when as we've said, we are warming the Arctic at rates about 2 times to 2.5 times greater than the rate of warming in the lower latitudes. And we looked at that when we talked about global weirding, and we talked about how the Gulf Stream and the jet stream are being affected by warming in the Arctic. Well, the other thing that's happening with warming in the Arctic is that gas hydrates that form in coastal regions of the Arctic Ocean and coastal regions of Antarctica, these are destabilizing. And all across the tundra in Canada, northern Alaska, Siberia, there are destabilizing pockets of methane being released. And so the methane is also a very potent greenhouse gas. And so the concern is that, there's this feedback mechanism which can accelerate the release of gas hydrate for decomposition of gas hydrate, and the release of the methane from that gas hydrate into the atmosphere, which accelerates further global warming and so on. And so that's a big concern for people trying to understand that. So, we can think about what gas hydrates are doing. There's this sort of short-term storage in the atmosphere. Carbon is stored in the ocean, the biota, and the sediments. And then long-term carbon is stored in land biota, like that, rock, soil, and what we call fossil fuels. So short-term in the atmosphere, short-term in a lot of the marine biomass should be considered short-term storage, and then medium-term carbon stored in longer-lived ocean biota, water, sediments, long-term carbon stored in land biota, forests, topsoils, and then rocks and fossil fuels. And so these 3 pools are interacting. And if we start moving some of the carbon from the long-term storage into the short-term storage, in other words, by taking fossil fuels and burning them up, we change this balance, and we drive that towards greater amounts of greenhouse effect, greater amounts of radiative forcing. So we'll talk more about climate change towards the end of the course when we're looking at hazards and so forth. So that's what I had to say about cycling. carbon cycling. We've also talked about nutrient cycling, and all of those processes are important for maintaining life in the oceans and on land, and for the long-term stability of the global ecosystem. So cycles are absolutely critical for that. And historically, you know, carbon dioxide levels have averaged about 300 to 325 parts per million. Geologically, if we go back and look at rock records or ice core records, we can see that carbon dioxide levels in the atmosphere have ranged between 150 ppm to about 400 ppm over the last 2.1 million years, what we call the Pleistocene. So right now, does anybody know what the concentration of these variations, or these different levels of atmospheric CO2 are correlated with different levels of temperature? Generally speaking, higher concentrations of CO2 result in higher temperatures. Of course, we can't resolve the exact relationship, you know, on an annual basis the way we can by measuring CO2 in the atmosphere today, but we can get a very general sense that CO2 is driving ocean warming, and there's good theory to back that up. We do understand how carbon dioxide and methane molecules behave in the atmosphere, and the sort of the basic atmospheric chemical physics of that. So in other words, more CO2, higher temperatures, less CO2, lower temperature. But why the high and the low and high and low? So there's negative feedback loops. You know, more CO2, more photosynthesis, less CO2. So that's a negative feedback loop. So as we increase CO2, you know, we would increase the ability of plants to take it up. And that's one thing that people who are skeptical about climate change say. They say, whoa, increasing the amount of CO2 in the atmosphere is good. It'll help the plants grow faster. We'll have, you know, more corn. So those are negative feedback loops. Another negative feedback loops is higher temperature increases evaporation, and more water. This should be a positive feedback loop. Higher temperatures increase evaporation, more water vapor in the atmosphere, more clouds. Well, you can also get more. You can get, you can have, this can work in a positive and negative way. So, carbon cycling is can be considered a closed loop, except that we've got these long-term storage pools, like fossil fuels, for example, which human ingenuity, human technology have started to perturb, and that's what's creating these conditions. Of course, it's also been what causes us to have, the kind of lifestyle that we do, and it's brought prosperity and high living standards to more people than have ever experienced them. So it's one of those trade-off things, but I think increasingly we're recognizing that the risks of climate change and warming due to CO2 are sufficiently high that we have to really treat this very seriously and look at changes. Okay, so that's as much as I wanted to say about CO2. There'll be more material in the readings. That's all I'm going to cover in the class for now, although I will talk more about fossil fuels and formation of fossil fuels and gas hydrates towards the end of the lectures. Now I wanted to talk about evolution. And we sort of all have an idea of what evolution is, but I wanted to, sort of cover some of the basics. And in particular, I wanted to look at the way in which our view of evolution and the way in which our understanding of how these evolutionary processes result in the incredible diversity of life that we have on the present-day planet, and how that's developed over 500 million, 600 million years of life history. I mean, there were single-celled organisms, probably 1 billion years ago, but only in the last 540 million years have we had multicellular organisms. And the way that those organisms have developed and the genetic material that's been passed from one to another, is what's given rise to this incredibly diverse, complex ecosystem that we presently enjoy. So let's try to understand some basic factors here. One of the things when we talk about evolution is the sort of convergent and divergent evolution. So an example of convergent evolution, so we start with jawless fish. Examples of jawless fish: lampreys, hagfishes, Amphioxus. These are all fish that are multicellular organisms that swim in the ocean that you would look at and say they're fish-like or eel-like. And then from these ancestors have come a variety of different groups, different classes in this case. And we'll talk what I mean about classes in a little bit here. So the class Chondrichthyes, which are cartilaginous fish, you know them as sharks and rays. And then from this original ancestor came land-dwelling stem reptiles, so ichthyosaurs, dinosaurs, and the surviving dinosaurs that we have on the planet today are birds. So birds are direct descendants of dinosaurs. Mammalia, we're all mammals, and we've derived from these different ancestors. So that's the divergence of evolutions from a single ancestor, multiple different pathways. So that's divergence from this single ancestry. However, the different organisms that are evolving in these different classes, if they live in the ocean, they face very similar sorts of requirements. They have to swim, they have to float, they have to capture prey. And the physics of the ocean environment, the temperature and mobility challenges that moving about and existing in the ocean environment are universal for, whether you're a shark or an ichthyosaur, a penguin, or a porpoise. They're all experiencing the difficulty of moving through the watery environment. And as a consequence of that, you can see that there's a sort of a general form that these different, very different, evolutionarily different classes of organisms have taken on. And this is what we call convergence in evolution. And so, they evolve this sort of teardrop shape because that's an efficient form if you're moving a large volume of organic material through a watery realm. And so, evolution both diverges and converges. It converges around common requirements, common challenges, and it diverges according to separation of individuals. So, convergence and divergence. I've also referred to diversity. And diversity, diversity now, when you say diversity, we understand that as being, the range of human beings, their racial, sexual, gender makeup. So when we think about, we want to have a diverse campus environment, we would think of people from many different backgrounds having many different appearances. Well, we can also apply this concept of diversity to the ecosystem situations. And so diversity has very specific sorts of meanings mathematically. So let's consider two hypothetical populations or ecosystems, A and B here. And I'm going to tell you that both of these populations have the same mix of species. And the mix of species, the number of species would be called the species richness. And so that's the variety that you could encounter within that population or within that community. But if you wanted to compare population A with community A with community B, you see that the diversity and the dominance is very different in these two environments. And so, how could we determine that if we were trying to, come up with some kind of a metric or a description of the relative diversity of these two different communities? So community one is clearly dominated by elephants. This is hypothetical, of course. You're not going to have a community dominated by elephants, but for the purposes of illustration, that's what we're showing here. Well, that's a And if you were sampling this population and you took random samples of, say, 5 individuals from this group of 100 individuals, how long would you have to take, how many samples would you have to take before you had found all of the existing species that can occur in this situation, in this community? Well, you know, it's intuitively clear that, you know, if we look at something that's relatively uncommon, like this critter here, you know, what this is supposed to be, a wolf or a bear or something like that, that if you were taking random samples from this community, random samples of 5, you'd have to sample quite a lot before you would find that, oh, lo and behold, this community includes wolves. So we would say that this is, you know, that the measure of dominance, you know, is much higher for elephants. And so, you know, on the other hand, if we look at community B, you know, we can see that we would, you know, if we were taking random samples of 5 or 6, we would much more quickly get to a more complete list of all of the different critters that were living in the community. So we can see that in, you know, in this community, we've got a single, I believe a single, and this is the second one here. We've got a single example of this, I'm going to call this a wolf. So we've got 2 wolves in amongst this 100, this community. And here we've got multiple wolves. So if you were trying to measure, trying to find out all the different species that occur in this community, you would much more quickly get a high number, and so that would tell you that you know, that the dominance of elephants is less in this community. And this can be important because you know, A and B, and, you know, if there were a disease or you know, some factor that was preferentially attacking elephants, this community would be much more susceptible to that than this community here. Now, there's a whole mathematics and a whole understanding of richness and diversity that's a little outside the, you know, the scope of this this course, but I wanted to introduce this as a notion because those of you who go on and take more biology and ecology courses will certainly encounter that concept. Diverseness and richity, you know, and this arises from, you know, both the evolution of different species and life forms, and then also from the environmental circumstances in communities of organisms that form. Obviously in this community, circumstances are very preferential for elephants, and so that's why we have this big dominance of elephants. Either that or elephants are killing off everything else, leaving themselves behind. Okay. And that can mean that, you know, that there would be a relative vulnerability of the 2 communities if there was some a process that was preferentially attacking elephants. This is, again, a very hypothetical example, but you can think of it in terms of, you know, a forest ecosystem. If it's dominated by 1 tree species, for example, and, you know, some disease that attacks that tree species comes along, and that's the dominant form, well, then there's going to be a major change in the environment. And, you know, in forest science, you know, across, you know, in the United States, North America, we've experienced that a couple of times. You know, there used to be big chestnut forests. So chestnut was a climax species, one of the species that, you know, that formed large forests. And they used to be a very important tree. When the colonists came over, they all used chestnut both as a source of food. They ate the chestnut nuts. And then they also used chestnut as lumber for building furniture. And you can find beautiful furniture from the colonial periods made of chestnut. Well, chestnut was wiped out by something called chestnut blight. We've had a whole series of blights and insect attacks. So right now we're experiencing up and down the East Coast the emerald bark borer, which is killing all of the ash trees. And so ash has been also a very dominant prevalent species, and that's essentially being wiped out by this disease. And so the richness and the diversity of communities has a big impact on how stable they are in the face of different kinds of challenges. All right, so we've been throwing these terms around, and now we want to get into some of the specifics. Hopefully this is review that you've had a lot of this in basic biology, but I want to put it in a marine context, and so I'll be using a specific example. So all life forms scientifically are described at some level or another, and we can come from a general description, which would include many different species, to a gradually increasing level of specificity, more detail, and greater discrimination among the different types that are there. And so this process, scientific description called taxonomy, is highly formal. So it's done according to a whole series of very specific rules. Because if you identify something as a member of a family or a genus or a class, you want to unambiguously place all other members of that family, genus, class, or species together with the organism that you've described. So the process for describing taxonomy is highly formal, and it has to be reproducible. It has to be done the same way every time. The process here was developed, scientists have been classifying the types of living organisms going all the way back to the Greeks. But it really was developed into its current form by the Swedish botanist Carl Linnaeus. He lived in 1770, before the formation of the United States as a nation. He died in 1778. But he started out, he was a botanist, and he made very careful drawings and descriptions of the morphology of plants, starting with flowering plants. And so he came to understand that if he described flowers, the morphology, the shape, the form of flowers in specific detail, he could distinguish among roses and pansies and dandelions, etc. And so he developed a system for doing that. And which we now refer to as the taxonomic hierarchy. Well, Carl Linnaeus was working in a time when all scientists used Latin, and so Latin remains the language of taxonomy. Now, of course, we don't speak Latin anymore, or most of us don't speak Latin anymore, but Latinate forms persist. And so when you describe a new species, if you want to give it a name, you try to make it into some kind of Latinate form. How does this work? All right, well, let's have an example. So we're going to go down the different steps here. And so these are the 5 groups or 6 groups of living organisms. And there are bacteria, archaea, protista, fungi, plantae, and animalia. Now, we'll see that there's a lot of problems with trying to distinguish here at the margins, but let's accept this for the time being. So bacteria, archaea, and protista, they're all single-celled organisms, monera. Then you get into fungus and plants and animals, multicellular organisms. And so if we're trying to step through, we're trying to go through the most general description all the way down to a very specific, and I use that pun intended, description of, we're going to end up with a soul down here. So what are the stages in all this? This soul is an animal, not a plant, a fungus, protist, or anything like that. So we can say that it's an animal, and it's in this kingdom. Within the kingdom, Animalia, there are many different phyla, and the phyla include vertebrates, Euro and Cephalochordata, and there are other animals, of course, that are, lack backbones. So Cnidaria, Mollusca, Annelida, and now, and so forth. So there are many, many different types, but the type of animal that we're describing has a backbone, has a vertebrae. And so, that's the next level here. From there we go into subphylum Vertebrata, because in this group Chordata, we also have the Uroencephalochordata. So these are primitive, less evolved forms of organisms that have a central nerve core that runs the length of the body. And then the vertebrae are organisms, vertebrata are organisms that have a bony encasement on that nerve cord. And these include birds, fish, mammals, reptiles, amphibians, agnatha, condyctes, so sharks and rays. And these are the boneless vertebrata, lampreys and hagfish and so forth. Okay, well, this subphylum is in the form of the fish, the Osteichthyes, and so here's the class of Octhichthyes. So within the class of Octhichthyes, you see how we're getting more and more specific as we go along here. We have a whole variety of different orders. And these are Clupiformes, that's the herrings, Gadiformes, that's the cods, Pierciformes, that would be things like rockfish and the red snapper, Pleuronectiformes, Lophiformes. Well, Pleuronectiformes, flatfish, is the best descriptor for soul. So we step down into families. So the order, from class Undictes to the order Pleuronectiformes. And within the Pleuronectiformes, there are a variety of different flatfishes. And the particular family of flatfish that the Rex sole belongs to is a Pleuronectidae. So that's the family. And then finally, from family, we step down into very closely related but still distinct species that include, two different kinds of flatfish: the witch flounder and the Rex sole. So the description of the Rex sole in scientific terms would be Glyptocephalus zechirus. And so that's how we get to that specific description. So all of that information is encoded in this description here, and has to unambiguously sort into this hierarchy of increasing specificity, starting with whether it's an animal, a plant, a fungi, and so forth, and then all the way stepping down to unique species. Well, what's unique about a species? Species are organisms. Species are life forms that can reproduce and produce fertile offspring. And so, that's can be crossbreeding and hybridization, and there are many different ways of exchanging genomes at this level. But species is one in which the genetic material is passed between and produces viable offspring that resemble the parents. So that's the classic view of the taxonomic hierarchy, and people talk about King Peter came over seeking from Germany, seeking fortune, I guess. I can't remember the Memnic now, but kingdom, phylum, subphylum, class, order, family, genus, and species is the order of the taxonomic hierarchy. And so here are is a description of these basic classifications of life. Sort of the prokaryotes, these are single-celled organisms and they lack a nucleus or other internal structural subdivisions. And so that describes the bacteria and the archaea. They have a single chromosome. Their reproduction is asexual. There's a huge diversity of bacteria. There's no nucleus or cytoskeleton. You know, and then, so that's one of the forms. Archaea, which weren't really discovered until the 70s, I guess, superficially similar to bacteria, but with many different sets of genes, different enzymes, capable of living in a very extreme environments. So the archaea are often referred to as extremophiles, organisms that like extreme conditions. So these are the prokaryotes. Now, of course, we can't, and these organisms have in common that genetic information that they contain is passed along in the form of DNA. And of course, we're all experiencing another life form, which has been very successful with the past year, and that's the life form of viruses. Viruses are even more simple than you know, these and they would fit into a subclassification. There's a lot of debate about whether or not viruses are alive, and you know, they certainly have the capability of reproducing. But they lack DNA. They're, you know, they're basically just a little bundle of RNA with some enabling proteins. But they can be very successful and have a huge impact. Okay, the other, you know, the groups that we care about, Protista, you know, these are unicellular, sexual or asexual reproduction. There's a lot of genetic diversity. And we are interested as oceanographers in Protista because diatoms, dinoflagellates, Radiolaria, Foraminifera, coccolithophores, these are usually single-celled organisms, and they're responsible for something like 95 to 96% of the primary productivity in the oceans. So the Protista are very important in terms of ocean productivity. Fungi, Plantae, and Animalia. So these are the, you know, the basic kingdoms of life, and viruses fit in somewhere here, depending on if you're a virologist or not. So, you know, we've got these different kingdoms, and if we think about, you know, the way in which these have developed, it sort of gives us an idea of, you know, the evolutionary history of life on Earth. And so this concept, this, you know, relates in trees. And, you know, evolution is often represented in this tree-like form. And I'm going to step through that in the next slide here. But just to introduce the concept of tree, which I'm almost certain that you've seen before, but it's a really important way of thinking about the evolutionary history and the relatedness of different forms. So in the kingdom here, the way that this relates is different groups branch off from common ancestors, and then within these branches there are other branches, and, you know, working their way down from kingdom all the way down to the species and genus level, you know, within these branches. And so the closeness to the branch point indicates the relatedness of organisms. And so bacteria, so the earliest cells that were capable of passing DNA information along, separated into bacteria and archaea. And archaea separated from this main group later in evolutionary history than bacteria did. And so archaea, in this presentation, are considered to be more closely related to multicellular organisms than they are to bacteria. In fact, the differences between archaea and bacteria are greater than the differences between archaea and eukaryotes in many ways. We can, follow this over here and look in more specific terms. So here we are in the branch of Animalia, and you can see that different groups branch off at different times, and so the where this branching occurs is an indication of how primitive the life form is. So one of the most primitive, and we use this word not in terms of a pejorative that is, represent things, but to describe how old or how the antiquity of its evolutionary divergence from the mainstream. So sponges, are considered a very, primitive, a very basic form of life. And so they developed early, and then the protistas here, dinoflagellates, silicates, amoeba. These are single-celled organisms, and then a sponge is a multicellular organism. It splits off, nematodes, arthropods, and you can see the divergence here. And so, at the top of the tree is, represented in, the most recent development in evolutionary history, which would be, the urochordates, and then finally the vertebrates. And we already looked, and we went through our description of the Rex sole, how the urochordates were, were an older form of organism that have a single nerve bundle that we refer to as the nerve column. Okay. All right, so that's the basic presentation of the tree of life. But there are problems with this tree of life, and trying to understand, how these different things came together. And, so one of the most important insights was gained by this woman, Lynn Margulis. And Lynn Margulis is kind of a controversial figure. She had a lot of different opinions. She was kind of very forceful in the way she presented with things. And she very early on came up with the notion that she observed that, in green plants, for example, that within green plants, what gave the green plants their form were these chloroplasts. And when you look into the morphology of chloroplasts and the organelles and so forth within chloroplasts, it was apparent to Margulis that these were very similar to free-living protists that you could find elsewhere, like cyanobacteria. And they had the same chlorophyll pigment. They had similar organelles, and she speculated that, but they're, a permanent part of plant cells. So if a plant cell divides, the daughter products from that division are going to contain chloroplasts, and chloroplasts, and the specific form of chloroplasts are a component of all cells in any reproducing species of plant. But Margulis proposed that very early in evolutionary history, perhaps there was some process by which bacteria, cyanobacteria, or organisms like cyanobacteria, entered into a partnership with some kind of an animal that allowed them what we would call symbiosis. And that over time, this symbiosis became a permanent component. The thing about symbiosis is that symbionts can live independently of each other, but chloroplasts cannot live independently of the plant cells that they occur in. And if you remove plant cells, chloroplasts from plant cells, then they're no longer able to exist. So Margulis's idea that the development of these vital organelles was a result of endosymbiosis, symbionts that were incorporated very early in evolutionary history, was, most people believe that now, most scientists. This has really sort of become accepted theory. But when she proposed it back in the 70s, it was extremely controversial, and she was, had a lot of trouble fighting the scientific hierarchy. So one of the people that changed that. So let's, it's worth, looking back on that and thinking about how our understanding of taxonomic trees has changed over time, it's worth looking back on this concept of the tree. And so what I've done in this slide is to gather a bunch of different pictures of these evolutionary trees. So I guess what I'm saying, the point I'm trying to make is that how do you draw an evolutionary tree where we assume that things branch off and diverge and don't come together again? So that, that sort of process there, that would sort of imply that these chloroplasts at some point or another got reincorporated into organisms that had evolved separately. And so, the original notion of trees, that was forbidden. That couldn't happen. And so, what's that saying is that these evolutionary groups evolve, and then they're taken up at first as symbionts and then eventually as organelles, and become part of species that have a much more extended evolutionary history. And so that's not something that Darwin thought about when he came up with the concept of the evolutionary tree. So this is a drawing from Charles Darwin's notebooks, circa 1837, in which he proposed that this is the way that individual species diverged, divergent evolution diverged from these common ancestors. 1837. Does anybody know when The Origin of the Species was published? There's a trivia question for you. Well, it was published in 1859. So already in 1837, Darwin was thinking about these processes, we believe, and came up with this concept of how the branching of evolution, the radiation of evolution, ramification of evolutionary groups would produce this tree of relatedness. So fast forward from 1837 to 1878, and this concept. So by that time, 1878, so Darwin publishes his Origin of the Species, and in 1859, there's great controversy. It's backed up. Other people defend him. Huxley defends him. There are other groups, and so already by 1878, the theory of evolution, even though we were still very shaky about the processes that allow evolution to occur, that's become accepted by scientists. And so Erwin Haeckel is a German philosopher and scientist, and he's working in the late 1870s. And so here's a tree from Erwin Haeckel. And of course, he's placing man at the apex of this tree, human beings at the apex of this tree, and then these more primitive life forms going all the way back to, amoeba and plantae, egg animals. So he's got a lot of things on here that groups that we don't think of any longer. Egg animals. I'm not even sure what an ovularia is. Plant animals, zoophytes. So this is a way of organizing different life forms that is similar to what we think about now. So he's got lizards, reptiles, birds, tortoises, crocodiles. He sees that birds and reptiles are related, but actually, they're so these trees are not correct. But in any event, this concept of the tree, becomes part of our understanding. Okay, well, we go from Haeckel in 1878 to a guy named Carl Woese in I guess 1978, more like 1972. I think it was Carl Woese was working. So Carl Woese is a kind of independent guy. He starts out at Woods Hole. He's working, he starts out in Indiana. He ends up working in a lot of different places, and he gets the idea. So all this evolution and all these trees, they go back to the process that Linnaeus used. And maybe I should have made this more clear when I was talking about Linnaeus. So Linnaeus based his specification, his distinction, his classification of organisms entirely on the morphology of the organisms. And specifically, he looked at flowering plants. And so the morphology of flowers, was, what he gained early insight into how they were related. Well, that thinking that you can describe species uniquely, if you're just careful enough about your description of their morphology, that is powerful. And that, because of course a bird, is different from a lizard in very specific ways. And if you just, if you're rigorous enough about your description, you can come up with descriptions so that you're not going to confuse birds with reptiles. Even when birds, as we talked about in the concept of convergent evolution, even when birds like a penguin resemble superficially a shark, we know that they're, completely different. So the morphological description, they have feathers, they have other, skeletal structures and so forth. So, all of this history of these trees, from Haeckel all the way through the early 20th century, they're all based on the morphologic descriptions or morphologic classification. All the classification is based on morphology. Well, this changes, with the discovery of DNA and RNA, by Watson and Crick, and then by others. And so, by the 1960s, scientists understood very clearly that the genetic code, the information which organisms during reproduction use to create copies of themselves, that information was embedded in the RNA and DNA components in the organisms. Well, Carl Woese was interested in distinguishing among different types of microorganisms that didn't have clear morphology. So he said, with your, there's a lot of, and when you're trying to describe, bacteria and distinguish one form of bacteria from others, one form of single cellular organisms from others, there's not a lot of morphological detail to work with. And so, microbiology lagged way behind. People recognize certain forms because they have distinct shapes or morphologies of rods and cones and different shapes of bacteria, different shapes and so forth. And so there was certain morphologic details, colors, things like that. But Woese was very unsatisfied with this. And he said, how about instead of using morphology, why don't we look at the basic information, the coding information that the reproduction of these organisms is using to reproduce copies of itself. So he found a section of the RNA, so that DNA separates into two strands, and the RNA copies particular pieces of genetic information out of sections of that DNA, and then carries it into cellular metabolism. And so one of the components of the RNA is the 16S RNA. And so this is a very robust component of RNA. And so Woese thought, well, how about if I just compare the 16S RNA of different life forms and see what kind of tree-like, what kind of relationships can we see based on this particular component of the genetic material. And so he started using not morphology, not the shape of skeletons or the shape of flowers, but the specific characteristics of the 16S RNA in different life forms. When he did that, he was able to reproduce a lot of the familiar tree-like branches of evolution, plants and animals, they all sort of, broke out. But the real shock for Woese was that he discovered this whole entire kingdom of organisms that were single-celled, but had a distinct or, they're single cell like bacteria, but they had a whole variety of different characteristics. And so Woese, using 16S RNA, was able to describe this whole kingdom of organisms. Well, Woese was, Woese worked closely with Linnaeus. They were colleagues and sometimes rivals and sometimes colleagues. And his thinking about, using taking the 16S RNA of the chloroplasts was a way to make a link from the chloroplasts in green plants, which evolved much later than earlier forms, and trace that back to cyanobacterial ancestors. So that linkage there was one of the lines of evidence that confirmed Lynn Margalis's thinking. So Woese, really turned over this concept of the tree of life. Well, in the decades since Carl Woese, the field of molecular biology has just exploded. And so everybody is using different fractions of genetic code to describe different organisms. And one of the shocking things, or one of the amazing things about the COVID-19 pandemic is the way in which the technology of identifying and classifying genetic material, specifically RNA, has exploded. And now it's something that can be done routinely. Back when Woese was doing it, it was very laborious and there were limited techniques. And nowadays, it's pretty automated. So, one of the amazing things about the COVID-19 virus is that it was sequenced. In other words, all of the genetic material, all the genes in the RNA of the COVID-19 virus was synthesized and sequenced on the 10th of January, 2020. A few weeks after it became apparent that there was this new potentially pandemic virus. And who sequenced it? Well, the Chinese. And so we call it the Chinese virus pejoratively. But we're fortunate that the Chinese scientists were able to sequence it so early because they shared that RNA. And that's one of the things that's happened, is that there's this vast library of all the different DNA and RNA sequences that any scientists have come up with. They're all put out there in the public domain. None of this is secret information. And so the Chinese scientists shared the RNA sequence of the COVID-19 virus on the 10th of January. Well, by the 18th of January, scientists at BioNTech and Pfizer, and then eventually Moderna, had taken that little bit of RNA and figured out that that the spike protein, the little lump that you see, those little horns that you see on the, all the pictures of the virus that we've seen, they figured, well, this is unique enough and this is important enough for the function of the virus because it's the attachment mechanism. So if we can make copies of that protein, then we can introduce that into the body, and we can teach the antibodies, the body to recognize that protein and produce antibodies to it. The immune system responds to that. And so, where we stand today with, potentially having, fought off this virus and getting back to normal life, we hope, that, owes very much to this sort of early work. Well, this concept that there's horizontal transfer of genes within bacteria and archaea, that's something that, is now being shown. So Palmgren and others in 2017 were able to show that, bacteria and eukaryotes, that this idea that once you've separated into a specific species, that there's sort of no going back, that doesn't really hold up, that there's a lot of transfer back and forth. And so this concept of the tree sort of breaks down, and, we have to come up with a new concept. And so, the idea of, the representation of the tree, has now got a very different form, from what Darwin was describing, almost 200 years ago, 180 years ago. And so this is the, bacteria, which are this hugely diverse group, and they've branched off from other single-celled organisms. So their archaea are more closely related to eukaryotes. We're all eukaryotes, all of the multicellular organisms. And so this branching occurred later. And so this kind of a representation shows us. And so now if you go to any talk that a microbiologist will give, they're presenting these very complicated trees of relatedness between different bacterial life forms. And, a lot of times we don't understand. We can see that there are similarities in the genetic makeup of different bacteria, but we don't know what those similarities do. We don't know necessarily what the different genes, what functions they have, if any. And so the understanding, we have sort of more information than we know how to analyze in terms of all that. Well, that's a long sort of discursion on that. I'll stop for a second and take any questions. I know I've been going on here, and but I wanted to presented a lot of material about evolution, so I want to talk a little bit more about how the way in which life forms have to adapt to a changing Earth, and how evolution can occur over different timescales. But let me stop and solicit questions or discussion about information that I've given you. Is this stuff that you know, or is this new, or is this clear? Or please raise your hand. Okay. Clear to me. Okay. Well, hearing no questions, I will carry on. So this is, where our understanding of life and evolution has, made great strides, and we see these trees. It's, a tree doesn't really work anymore. It's kind of like the branches can come back together and form new branches and separate again. And so the ability of life to adapt to these different circumstances is very impressive. And in terms of the coronavirus, which is affecting so much of our life, we're now conducting this amazing experiment in how an evolving virus will respond to some of the pressures that we're putting on it. So the virus is driven to reproduce, and it's sort of a very simple evolutionary formula for viruses. If you are successful and you can invade a human host and command that human host to the cells in that human host to stop reproducing cellular copies of itself, and instead make millions and millions and billions of copies of the virus, then you're a successful virus. Your variant is successful. And so what's happening now is that we've got such a huge pool of a virus. It's so many different variants of virus that are out there, that it provides, that there's enough sort of variation in the reproduction of, the reproduction is successful, and you produce exact copies 99.99999% of the time, but every once in a while there's a divergence from that. And if those divergences happen to infer or confer an extra ability to evade vaccines or to attach more successfully to lung cells, well, that variant is going to explode in prevalence. And so that's what we're facing right now. We've got this, huge pool of infected people, and the viruses have the ability to multiply. And in specific urban areas where there's been a high prevalence of bacteria of the COVID, we're seeing the emergence of different variants. And so there are New York variants and variants from Brazil, South Africa, Great Britain, any place where there's a big center of a virus. You get these new forms emerging, and the concern is that some of them might be more lethal, some of them might be more transmissible, some of them might be more able to overcome the antibodies and immune system that's primed by the use of vaccines. So, we're sort of facing a real-time, once-in-a-lifetime experiment with how evolution works. And we're very, very lucky that we have these tools, which were developed without thinking about, this possible application. But these tools and this understanding of how evolution and genetic code works are what are enabling us now to have a chance at fighting this thing off. Okay. All right. So, here's the coral of life. This is the idea that this diversion. And so actually Darwin drew corals as an example. So Darwin is always ahead of all of us. All right, so let me stop there with evolution and talk about how different events in Earth history might have driven different levels of evolutionary occurrence. And we're most interested in life, and we have the best understanding of multicellular life that's existed over the last 550 million years. And so that's the period referred to as the Phanerozoic period, and that's the period of high diversification, of huge ramification of life forms, and of an alteration of the Earth system by the interaction of these life forms. And so geology and biology work very hand in glove in terms of presenting different opportunities for stable communities to occur. So we can imagine that there's been some period of 10 million years, 100 million years, and a community of organisms has established itself. And there's dominance and hierarchies, and those organisms are it forms a sort of stable ecosystem. Well, we know from geologic exploration that there have been various times in history when catastrophic events, have wiped out large numbers, large percentages of the living life forms in any particular time. And these, the largest of these events are called the extinction events. There are 6 great extinctions. There are 6 in this species. And so what this graph is showing us is the percent extinctions. And so the earliest one that we know of occurred in the Ordovician about 435 million years ago. So that was a time when life had existed for approximately 100 million years or so. And there were diverse forms. There were sort of fish-like organisms, and some simple plants and mosses. Flowering plants had not emerged. And then there was some event that probably sort of large-scale volcanism that wiped out great many species. And so these events, when that happens, when the slate is wiped clean, that provides an opportunity for new life forms to evolve. And with the evolution of new families and new life forms, those life forms can subsequently evolve. And so these great extinction events, although they're very bad news for the organisms that are alive when they occur, they create a sense of opportunity, or a situation of opportunity for further evolution. And so these extinction events, the Ordovician, the Devonian, the Permian, the Triassic, this is the one that, that people most often focus on. This is the Cretaceous events. So the Cretaceous is a time when that was the sort of the time of dinosaurs. And dinosaurs, saurids generally, were the dominant life form. And one speculates that if, and so 65 million years ago, there was a comet which impacted the Earth, and there's controversy about what exactly, how much of the, well, then that led to, after that point, we don't have dinosaurs anymore. And there are other explanations, and there are other things that may have worked together, but certainly the impact of this large comet with Earth was very, very bad. And there are fossil remains now. It created a huge tidal wave, and so there were the waves from the Gulf of Mexico, then Gulf of Mexico 65 million years ago. Waves from that impact penetrated as far as Montana, Wyoming. So there was a huge surge of water, and there was a massive impact to the atmosphere. And so, in an instant, life became untenable, and thousands and thousands of species were wiped out, including most of the large dinosaurs. The birds, which eat seeds and which can fly, had some advantages over organisms like tyrannosaurs, T-Rex or whatever. And so, they were able to escape some of the worst conditions, and they were able to find food because seeds are preserved. And so the speculation is that that form of the Aves form, continued. So, these events, these catastrophes, provide the opportunities for new evolution. And sort of the, the late Eocene, 35 million years ago, there was a lot of large-scale organisms that were lost. But some scientists, refer to the present day as being the sixth extinction, because we're wiping out, species are disappearing due to the changes that human land use and human climate change and other processes driven by human society. We're wiping out species at a rate which seems to be comparable to any of these earlier events. And so we're extinguishing a lot of species, and those extirpations go to a very deeper evolutionary level. So these extinctions occur. So that's one of the things. Any questions about this? Okay. Well, other processes, temperatures and dissolved gases. These are conditions that life has to face, specifically in the ocean. So we already talked about the presence, the solubility of different gases. Oxygen and CO2 are relatively soluble. CO2 is the most soluble in seawater. And so the ocean has got very high concentrations of CO2 or CO2 and bicarbonate compared with CO2 in the atmosphere. So the oceans absorb a lot of oxygen, and the availability of dissolved gases is a sort of critical function. Nitrogen we've talked about, because gaseous nitrogen, which is the most prevalent form in seawater, is not bioavailable. It can't be used until the nitrogen is split and fixed. Other processes that organisms living in the ocean have to deal with are the osmotic pressure, because the oceans are salty. We've talked about salinity in different terms, but organisms facing salty, living in a salt environment, have to maintain a, their different. They have to maintain their internal cellular salinity, osmosity, has to be maintained against the gradient in the outside world. And animals living in a fluid media have to either have the same osmotic concentrations as the surroundings, or do work to keep the balance. And so freshwater organisms, for example, fish living in freshwater, their internal, the osmotic content of their internal cells is has a higher gradient than freshwater. And so to stop absorbing, so freshwater wants to permeate the cellular membrane to osmotically enter the cells, and so freshwater fish are constantly trying to pump out water in fresh water. So living in, whereas organisms, marine fish, have roughly the same osmotic pressure intercellularly as they do in the ocean. And so it takes more work for organisms to live in fresh water than in salt water. If you put a freshwater fish into salt water, it dies pretty quickly. But if you take a saltwater fish, and it can adapt to living in fresh water. Okay, well, it looks like I'm out of time. Let me just tell you that I've found a series of videos, but not everything that's I'm not going to have time to cover everything in lectures that I would like to get through. And so I've found a series of videos, and I'm going to assign these as homework. And so you can watch these videos and get some basic information, and then there'll be a little reading quiz or video quiz to go along with those. They'll be worth 10 points each. And I think we have we're behind a little bit in the number of quizzes that we've given. And so I'll be putting those out in addition to the reading quiz. Okay, any questions? You have been very quiet. No questions. All right, everybody. Well, thank you for attending today, and I look forward to talking to you again next Tuesday. And of course, we're going to have student conferences on Milestone 3. They've been set up by Carrie, and your different teams can sign up, and we'll all get together. Okay, bye, everybody.
Thank you. Have a nice day.
Thanks.
So, are these, sorry if I missed this, if you had said it and missed it, but are these extra reading quizzes for homework this weekend, you're saying?
There'll be one, yeah, I'm going to assign you a video on primary productivity, and that'll be due this weekend. It won't take long. It's about a half an hour to go through, and then the questions are actually embedded into the exam. But I think that the combination of watching the video, it's a nice video, and then responding to the quizzes, the questions will help to cement the material, and that'll help me cover, give you a chance, give you an exposure to problems about primary productivity. So yeah.
Got it. Thanks.
Sure. Okay.
Again.
Any other questions?
I have a question. It's not really about like the lecture though. It's kind of random.
Well, I'll answer them. What the heck?
Okay. So I signed up. You're listed as a professor for next fall for OCE 4017.
Yeah.
So my advisor said to confirm confirm the prereqs. So I was just wondering if there's prereqs for that class or like what they are.
No, I mean, if you've survived oceanography, you'll survive 4017. 4017 is, has a more of a discussion format, and we branch out more widely into, into sort of, it's issues in environmental science is the formal name of the class. And, the two classes, oceanography and issues in environmental science, work kind of well together. So I have a lot of students that take both. And so, but we'll be doing a lot more policy and a lot more, not just marine processes, but also looking at agriculture and that sort of thing.
Okay. Thank you.
Sure. Okay, bye.